This immune response uses T lymphocytes,
which is produced in the thymus gland.
It is then stimulated to divide many times,
forming clones.
These circulate in the blood.
Until it meets an antigen it has the receptor site for.
There are 3 types of T lymphocyte.
Killer cells, which break up target cells, therefore destroying virus and cancer cells.
Helper cells activate B lymphocytes to produce antibodies. However, cell-mediated immunity
is an immune response that does not involve antibodies,
but rather involves the activation of phagocytes, antigen-specific cytotoxic T lymphocytes, and the release of various cytokines in response to an antigen.
An example of cell-mediated immunity is the rejection of transplanted organs as a result of the antigens being recognised as non-self.
The body responds to antigens outside the cell.
In humoral immunity,
Antibodies are produced with special properties,
such as being able to dissolve in blood and tissue fluid but not attached to cells.
B lymphocytes are a type of cell in which millions of antibodies are binding to the cell surface membrane,
When an antigen
such as fungi or bacteria.
enters the body, the B cell engulfs the antigens and processes them as APCs.
That stands for antigen-presenting cells.
Once the APC is formed, activated T helper cells combine to this B cell for clonal selection.
At this point, the B cell with the correct antibody is chosen for cloning.
The interleukins produced by the T helper cells activate the B cells
This causes the B cell to divide by mitosis.
to give clones of B plasma and B memory cells.
This is clonal expansion,
These plasma cells then produce antibodies.
which target antigens at the surface of pathogens and disables them.
These can take weeks to be fully effective against a pathogen. This is why when we are ill, the symptoms are a result of the pathogens dividing freely.
Some of the clone B cells develop into memory B cells.
If the body is infected by the same pathogen, these memory cells divide to produce plasma cells.
They then make the right antibody and wipe out the pathogen quickly.
This is called the second immune system.
It is then stimulated many times.
You've got to divide. Do it again. Do it again now. Do it again now. Do it again now. Quickly.
About to get to toll off your chest, ooh. I feel the tears and you're not alone.
Okay, wait, can we do it again with Harsh?
No, it's fine.
He's smiling at me.
Okay.
Just leave Harsh, just...
For what they're selling, I'll send you.
Also known as antigen present. I said antigen, not antigen.
In humoral activity.
Immunity.
Oh shit. In humoral. In humoral activity. Oh shit.
Be a little more easier. So what you've done is you said that hard cells such as fungi and bacteria.
Once the B helpers. Oh shit. The interleukin produced by the B helper cell. Oh, carry on.
How long do you reckon you'll be finished?
5 minutes. We can give us 5 minutes, alright?
Yeah.
Okay, well, we'll leave you alone.
Cheers, bro.
Yeah.
These can take weeks for it to be fully effective against a pathogen. Oh shit. If the body is infected by the same pathogen, these memory cells. Okay, I got it. If the body.
Okay.
If the body is infected by a new pathogen, these.
Same pathogen.
If the body is infected. Sorry, I'm trying not to laugh. If the body is infected by the same pathogen, these antibodies divide to produce new. Hey, these memory cells divide to produce what? Plasma cells. Oh, just plasma, there's no new.
Plasma cells.
Okay.
Plasma cells.
If the body is infected by the same pathogen, these memory cells. This is called the second immune system. Okay, get out this fire, I don't know why.
If you come with me, ooh, easy, and a 1, 2, 3, 4, 5, 6, 7.